I'm Eric Royer. I work as a swine nutritionist for the IFIP Institute in France. In the Feed a Gene project, I'm involved in the first work package 1. The goals of the work package 1 are to produce new innovative feedstuff, new processing of raw materials for monogastric utilization, and to evaluate new methods of assessment of the nutritional value of this feedstuff. I'm the coordinator of subtask of the work package 1. Precisely dedicated to the characterisation of soybeans and rapeseed. European soybeans were mainly used for human utilization, and soybean was an important feedstuff. Nowadays, the soybean production is expected to grow more and more in Europe. There is also a demand for using the soybean as GMO-free and locally produced material to identify and label this product as this, and to use it for animal production. It seems necessary now to work on expeller soybean meals in Europe because we have few data on this process. Crushing European soybeans in large crushing plants is very complex because it won't be easy to identify and to label the origin of the beans, either imported or either locally produced. So that will be easier to do in new plants of medium or small size, and for that an expeller process is more adequate. We have tried to work on the parameter setting of the processing of the crushing and of the oil extraction by comparing different temperature and different time of eating of the beans and choosing the optimal. That's the first way to do. The second idea was to test the effects of dehulling, of removing the hulls of the beans, in order to increase the protein content of the final soybean meal. From our test, we are satisfied with the result. They are similar to the value that we expected. And we obtained good quality products with good values for residual oil, krill protein, and protein solubility. So now two systems are available: extrusion or flaking cooking. For an industrial, for an operator, the choice can be made following his needs for the market. Extrusion has to be dedicated to the soybeans and cannot be used for other oilseeds. The flaking cooking has a lower gross margin per ton, but it can also be used for processing rape seeds or sunflower seeds. We hope that the next steps of our work will be to discuss or to help operators to work on a project of investment, and perhaps that we will need to do economic studies. At the end of the Feed a Gene project, we hope that the products we have worked on will be interesting and could be used by the operators, by feed manufacturers or other operators. This can help them to be less dependent of the important protein sources and to use with more accuracy the protein products produced in Europe.